Hello everybody, which everything is fine. We will talk now about lecture 4, editing and coloring using Jalview. In this lecture, we will see many things about how to fetch your references and for sequences, and also how to set your feature setting and change the view, and many other things we will see during that lecture. So simply in this lecture, we have to know the main 7 steps in Jalview. The first step is to import your sequence, either a drag, drop, or paste or URL, as we described in earlier lectures. Also, we will see how to can decorate your sequence with references, annotation, and from external databases. Also, create your alignment. And number 4, using built-in shading, conservation analysis, tree, and PCA capabilities to explore. PCA means principal component analysis. Maybe we'll describe that later in this lecture, not that lecture. Select regions for refinement or further analysis. Also import trees annotation created with other programs to explore further, as we'll see in this lecture. Also prepare annotated views for publication. As we have to remember now that Jalview as a multiple sequence alignment, just viewer, editor, and its analysis tool that can be used. Jalview is designed to be, as we said, it's an independent platform for running on, it can be, of course, running on Mac and Microsoft, Windows, Linux, and many others actually platform and that support Java. So this is the main important thing you have to know, that you have to, whatever platform you are using, it can be included, but it has to support the, Java. So we'll see, as we described in earlier lectures, that there's a multi-functionality for Jalview, and that's why it's one of the favorite programs that can be used by bioinformatician. And as we see, that can edit and analyze, of course, large alignment and with minimal degradation performance. So that's why I like to use it, and has many algorithms, multiple sequence alignment algorithms like cluster W, cluster omega, and also muscle, MAFT, TCAFE, GL props, propcons, and MSA props. So how and where we can find that these algorithms? Simply just go to web service menu here after uploading your sequence, as we have shown in the last lectures. After uploading your sequences, you can just go to web service here, this menu at arrow number 1, and after that you just can go and press alignment at arrow number 2. You will get this menu, sub-menu here, which have many algorithms to use. So as you can see, you can use any of these algorithms with default, like we show here at arrow number 3. You can use TCAFE with default, or propcons with default, muscle with default, MAFT, and others, as you can see, just with default. The same thing, as you can see here, is maybe here much more clear. Here is the menu, web service menu, alignment, and you can choose any of these algorithms by default. Also, you can use it if you try to make a preset and you already have a preset for this algorithm. You can do the same thing here also, as you can see from here, web service at arrow number 1, and then choose alignment at arrow number 2. And then from here, you can go to this sub-menu here at arrow number 3. And then you can choose, as you can see here, after making the preset, you can choose the preset one, the one that you already, if you change the setting for each of this program. So, okay, so somebody will ask me how we can change the setting of any of these algorithms. Simply, as you can go also from the same menu here, go to the web service, and then alignment, and then go to, of course, edit, setting, and run. At the same menu, you can also change the setting of the program. So the first choice is TCAFE with default, which means that you use TCAFE with default. The second choice is to change the setting, and the third choice, you can use the preset and run your program. So you can use the default program, you can reset the the setting of the program, you can, of course, use the preset as we said before. So for each, this apply for each of these programs that we mentioned earlier. Okay, this is of course the menu of presetting. You can see you can change any of the setting you want. Okay, now we come to the coloring, because of course after you make your multiple sequence alignment, you would like to use a useful coloring to interpret your data. So that's why there is many coloring scheme, as we said earlier, based on many things, as you can see here, and there is some for DNA and some for protein. The color scheme, as you see, you can choose color scheme is a matter of taste for you, and not just taste actually. I think it's also a matter of what you would like to show in your science and your multiple sequence, what you would like to show to the people in the paper, and which scheme is presenting your ideas that you would like to use. So how to go to the coloring scheme? Simply, you just open the color menu at arrow number 1, and you have many color schemes here, and you choose whatever you want. For example, you can choose cluster X, blossom, and there is differences between this color scheme, as we mentioned before, and maybe we'll come to that later also. So this is, for example, cluster X, one of the most, I mean, people usually try to use this one because it's easy to understand. And it initially, of course, designed by Toby Gibson at EMPI, EMPL, actually, European Molecular Biology Laboratory, and probably one of the most popular, as we said. Of course, this is depending on what you would like to show, as we said earlier. So this is one of them. You just can upload your sequence data. Jalview, of course, can read and write many common sequence format like FASTA and Clustal, MSF, as we mentioned before, and PAIR. So you can apply your own data and your sequences, and then you can, as we mentioned, make a multiple sequence alignment using any of the program we showed, and then you can color them using the color you want. One important thing that you would like to show here in this lecture, also how to fetch the database references to your sequences. If you have sequences and you don't have the database references for that sequence, so you can do that annotation retrieval and also other things using Jalview. It's very simple. You just upload your sequence like here, and then go to the web service at arrow number 1, and then go to arrow number 2 here, fetch database references. And of course, you can choose from this sub menu. There is many options you have. First, as you can see here at arrow number 3, trim retrieved sequences, or you can have your standard databases. You just grab the annotation and information using a standard database to fetch your database references, or from the choice here at arrow number 4, from MBIL to UniProt. This gives you actually like a range of databases here, as you can see. Here you can choose whatever one, which one we would like to use. For example, I can use, if I choose here at arrow number 6, UniProt, he will retrieve this DB references database from the UniProt database, if I just clicked at that one. But simply, you can do this for one sequence or multiple sequences like here. Here, I just go to the web service, do the same thing. After, of course, after importing many sequences, as you can see, and then I can just select them and then do the same thing. Open the web service window and choose fetch DB reference like here, and then go to standard database. In this case, he will retrieve and fetch the DB references for all these sequences. I already put them here, and this is very easy and very important. And at one step only, you can already retrieve the database references from different databases and then include it in your work. Okay, now if I would like to, of course, based on these references, I would like to color certain features. As you see, just go here to view, and to see this feature in my multiple sequence alignment, go to view here, of course, and then check, as you can see at arrow number 3, show sequence features here. You can just check here. Of course, you have to just check automatic scrolling. This is very important at arrow number 2, and also check show sequence feature. Here, you will not just see the colors, but also you will see different features, as you can see in your sequences, aligned sequences, these features with different color. As you can see here, for example, there is many features that can be detected from your sequences, and you choose whatever feature you would like to show, and each feature have a different color, as you can see here. So each feature has different color. What feature you would like to show in this sequence alignment, just press here and check here in front the box in front of the feature. For example, if you would like to show helices, where is the helices in these multiple aligned sequences? If I would like to show that the helices position are conserved and I would like to show them here, just check here, choose helix, and of course that will be the color, which could be changed also if you want. And then check here in front of that. So you will show that your helices throughout your multiple sequence alignment is conserved. If you want to show the conserved position or the position for the whether conserved or not phosphorylation, for example, for the Y amino acid, hydrogen amino acid, or serine or threonine, whatever, any one of these ones, you can choose them. For example, if you want to choose that one, so you this phosphorylation site, you can just will be marked with that color, and then you can just check here, check the box, empty box here, and say, okay, what will happen is all the position for that amino acid will be colored with this color, which could be changed if you want. So this will mark all that amino acids throughout all the sequences. So this is a very good thing to show if your study is, for example, concerning about this phosphorylation amino acid like Y, S, T. So any of them you can just show them across these sequences, and the position of it can be indicated by that color, as we said. Any of these features can be utilized with the same technique as we said. You just choose the whatever feature you would like to show in your multiple sequence alignment, and then of course the color is indicated, and also you just check in front of it and then press OK. Here, of course, there is one blank. There is this sequences of some proteins with no color, as you can see. So you cannot recognize any important thing in it. So you just have to wrap them first. As you see, you can wrap them so you can see all the sequences aligned from here, format, and then wrap. And then when you have them like this, you can after that color them, and of course do the same thing what we showed you earlier to put the features on this whatever feature you would like to do with these sequences can be just presented after that. So it's very easy, of course. So you can reorder after you're making your alignment. You can reorder your alignment. This is, of course, this is one example. Just I put my sequences here. This is before the alignment, and this is after the alignment. As we can see, after the alignment, some positions and some domains is already have been shown to be conserved, like this one in that box. So that's I just put here outline here. Try to outline that this domain is conserved in this between these different proteins from different species. So, and then I can color that also. I just mark them, but I can color them and reorder the sequence based on the multiple sequence alignment. So I can change instead of just putting my sequences randomly, and I would like to order them based on the multiple sequence alignment. So this can be done very easily by pressing calculate from calculate menu, and then press sort, and then from sort there is sub menu here comes by which way you would like to sort your alignment, multiple sequence alignment. So of course you have many options here at the sub menu number 3, at arrow number 3. We can reorder these sequences by ID, by length, by group, pairwise identity, or by multiple sequence alignment ordering. So here I just checked this arrow number 4. So I just order the sequences based on the multiple sequence alignment order. Of course, you can also order it simply by your hand. You can, if you want to change the order of these sequences here at the bottom, like here at arrow number 1, I just can click and just choose them and then change it and by the arrow key. So this is a very easy way to reorder your sequences based on what you would like to show first. Of course, you can practice and read this practical session by trying to on your own time, of course. Here is that's a link. If you just press in that link, you will get some multiple choice questions, and you can just practice with it. Thank you very much, and see you in the next lecture.